Now we will discuss methods of immunoprecipitation. Mainly there are two methods. One is direct immunoprecipitation and the next one is indirect immunoprecipitation. ये हम discuss करेंगे one by one. In direct immunoprecipitation, antibodies specific for a protein, they are coated on a solid substrate like agarose beads or magnetic beads. So coated beads with antibodies, they are used. The beads with the bound antibodies are then added to the protein mixture from where we want to separate a particular protein or antigen. Proteins or antigens which are targeted by the antibodies, they are captured onto the beads via the coated antibodies. As we have discussed, these antibodies coated on the beads, they are specific. Specific antigens, they bind with these antibodies. The proteins or antigens in the sample are then immunoprecipitated. This direct immunoprecipitation is quite a fast method, as there are less time required. Why less time required? Because there is less incubation required in this process, and it also reduces non-specific false positive results. So this is direct immunoprecipitation. Now we will discuss indirect immunoprecipitation. Antibodies that are specific for a particular protein or antigen, they are added directly to the sample. So they are not coated on the beads. Well, they are free, so they are added into the sample. Antibodies, as I have mentioned, are not coated to solid spot. Antibodies bind with their target antigens in the cell or tissue homogenate to form antigen-antibody complexes. And then protein A or G coated beads are added to the mixture so that this complex react or bind with those beads through antibody. And antibodies bound with their antigens, they get attached with the coated beads. Antigen-antibody complexes, they are separated then by centrifugation from the rest of the proteins or antigens present in the sample. जाहिर है beads होती हैं, तो जब हम centrifuge करते हैं तो beads नीचे sediment हो जाती हैं. So जब beads नीचे sediment हो जाएंगी tubes में, तो उनके साथ जो bound complexes होते हैं वो भी sediment हो जाएंगे, और जो rest of the proteins हैं, antigens हैं, वो ऊपर रह जाएंगी supernatant में. Supernatant को हम discard कर देते हैं. Both methods give the same result, as the protein or protein complexes which are bound to the antibodies are immobilized on the beads. This is common in both of the methods, I mean in direct method and in indirect method. Indirect approach is preferred as compared with direct approach, especially when concentration of the protein target is low. ये इसकी ज़ाहिर एक advantage है, or when the specific affinity of the antibody for the protein is weak. तो अगर उसकी concentration कम हो या उसकी antibody के साथ affinity कम हो, तो तब हम indirect method जो है इसको prefer करते हैं. ज़ाहिर है at the same time this indirect method is time taking, as there is more incubation and washing steps as compared with direct immunoprecipitation.